All right, Unit 4, Sensation and Perception, involves all of the sensory information that comes into the brain from the 5 senses on the sensory neural networks, and what the brain does with that information as it makes sense of the information and converts it into something that it interprets or recognizes. Pretty decent amount of information in this chapter. But we'll quickly go through the difference in the 5 senses, which is essentially what you would want to know for the AP exam, and then the different ways that the brain can be tricked when it's perceiving things. So starting with bottom-up processing versus top-down processing is one of those confusing pairs of vocab words that people tend to ask about. Bottom-up processing is going to be the analysis that begins with the sensory receptors and works its way up to the brain to be recognized. So for instance, this is going to be any new or unknown information that's brought to the brain from the outside world, whether it's a vibration of light, a vibration of sound, or pressure on the skin, anything that the sensory neurons collect from the physical world and bring to the brain. So bottom-up processing is raw sensation. Top-down processing is more of a brain first activity, hence the metaphor of top-down. Top-down processing denotes what the brain does when it perceives something in anticipation. So information processing guided by higher level mental processes such as expectations, prior knowledge, or any interpretation that the brain may make. Examples of top-down processing would be expectations that your brain has. When you go into a restaurant that you've eaten at before, your brain, and you order something off the menu, your brain is already creating an expectation of what it's going to receive as a taste or as a smell, as a sight. So familiar information to the brain allows it to top-down process, which means it's guessing based on past experience, and that's something that's beneficial, obviously, because it can save time. The brain can anticipate what it thinks will happen, which is something that can be tricky at times. When you anticipate that you will be in pain, when you are looking for something scary or dangerous like a snake while you're walking through the woods, or a scary man walking in the dark, your brain is going to perceive or expect that it's going to happen. So top-down processing is often explained using pain in FRQ questions. If you go to the doctor and you get a shot, you're anticipating that that shot is going to be painful, and it may actually increase the actual experience of pain because of your brain's expectations. So bottom-up processing is new information to the brain. Top-down processing is expectation that the brain has because of old information or experience. Threshold. So there's a lot of questions often on tests and retakes about the difference in thresholds, absolute thresholds versus difference thresholds, and things like that. So the threshold is the term that we use of the minimum amount of stimulation that's required in order for you to actually experience that sense. So if we're talking about the absolute threshold for sound, it is the bare minimum decibel that's required for your brain to actually comprehend the sound half the time that it hears it. An absolute threshold for physical touch or pain would be the minimum amount of pressure that's required in order to stimulate a sense of touch. Signal detection theory is this idea that your brain is better or worse at picking up or detecting, we'll say, sensory information based on several different factors, whether it's something that it's looking for or it's experiencing, something that's motivated to see. Again, that top-down processing of expectation. If you are, let's say, again with the pain, it's a good example of signal detection theory. If you're expecting pain, you're probably going to anticipate a painful feeling, and therefore it's going to be a lot easier for you to feel it when it happens because you're expecting it. So signal detection theory is this idea that your brain can actually create things or can actually experience signals from your sensory input even when maybe it's not necessarily a measurable amount. It's just essentially that your brain has created that. It's a form of top-down processing. Subliminal thresholds or subliminal messaging, subliminal touch, subliminal sound, anything that's subliminal, sub meaning below, that's below your awareness below your conscious level of awareness. So if it's a subliminal message, in theory it's a message that's below what you can actually hear, but your brain is somehow still detecting it. So if you buy into subliminal thresholds, it's this idea that something that can occur below our threshold. And in fact, we know this is true for sensory data, because there are sounds that we cannot detect. There is sight, light wave decibels or frequencies, not decibels, excuse me, frequencies of light that we cannot detect, and we know that those exist. So there are subliminal sensory data that the brain cannot detect. Maybe other animals can detect them, or maybe instruments can detect them. X-rays, gamma rays, we know those exist, but they are subliminal for our absolute threshold for sight. So subliminal is below. The difference threshold is a comparison. So a lot of times people get confused with the absolute threshold and difference threshold. What's known as the just noticeable difference, or the JND, is this idea that you can perceive something as it changes. So difference threshold is the amount that has to change in comparison to what it was before. And again, it's a comparative statistic. It's a comparative term. Weber's law is kind of an example of the difference threshold. It's the amount that something has to change on a constant continuum in order for it to be detectable. So for instance, if you were to cut 1 millimeter of length off of your hair each day for 6 consecutive weeks, it may not be detectable that your hair is different over time. But if you were to cut it by, say, 10% every single day, that's a constant enough percentage that it would be noticeable. A 10% increase in anything, or a 10% decrease in something, is going to be a noticeable amount. So Weber's law is named after the person who came up with it. Basically, that to perceive something as different, it has to have a constant detectable percentage of change, right? It has to be a detectable percentage. Now we get into the senses themselves. Let's start with vision. This is a chart that was included because people often want to know the order in which light passes through the eye. So if you look at, in order to this word transduction is the idea of taking a stimulus energy from the environment, from the world around you, and converting it into a pulse, changing it into an impulse. Your brain doesn't understand light waves. Your eye detects light waves, brings them as bottom-up processing to the visual cortex in the occipital lobe, and the brain makes sense of that as being something that has been transduced into a sight. Maybe it's a color, maybe it's a shape, maybe it's an outline, maybe it's a moving object, maybe it's something that's farther away. That's what your brain does with that information. So transduction is converting a stimulus energy, like a wave, into an impulse that the brain understands. So when you look at the pathway to the retina is actually where this transduction is going to take place. This information is going to be absorbed in the retinal cells by cones and rods, and it's going to be converted and travel down the optic nerve. So you have the cornea, which is the outer protection of the eye. You have eye juice inside the cornea, the moisture that keeps the eye lubricated. There's basically just a vacant opening there, a chasm. You have the iris of muscular tissue, which is the ring of tissue that gives you the color in your eye. You have the pupil, which is the opening, kind of like the window. The analogy that I gave in class was if your vision was a window into your house, then the pane of glass in the window would be the cornea. The iris would be the curtains that you draw to block the light coming in. The pupil is just the opening in the wall, on the inside of the wall. So that is the opening to the eye. It passes through the lens, where it is put into focus, shape and things come into focus through the lens, and then the light travels down to the actual retina itself. And the retina has a right visual field and a left visual field, and they come together and make up the optic nerve. And the optic nerve is actually what travels that impulse to the brain through the thalamus to the visual cortex on the back of the brain. There's a lot of information here. about the cornea, the pupil, the iris, the lens, everything from the sclera, the optic nerve, the blind spot, which is where the two retinas meet, the right visual field and the left visual field. There are no receptor cells there, so you have a blind spot. You also have the fovea, which is not on this sheet or not on this graph. The fovea is the optimal focal point where light is concentrated with the most light wave data in that spot, and that's the optimal viewing point for your vision. That's where your vision is the most focused. The retinal wall is made up of tissue cells, of cones, rods, ganglion cells, basically just the axons of optic nerve cells, and that makes up the optic nerve itself. So you can see from the retinal wall there, light with that yellow arrow is where it is actually absorbed. Then you see things like ganglion cells, bipolar cells, then the cones and rods, which the axons of which make up the actual optic nerve themselves. So rods are for black and white, seeing in low light, and even night vision. You couldn't see in complete darkness, complete absence of light, but you can see in low light. Rods also help in peripheral vision, which is the rods are concentrated away from the fovea, so light that enters in through the pupil and goes to the extremes of your visual fields, the extremes of your right visual field and your left visual field, the outer edges of your retinal wall, we'll say. Those are going to be on the periphery of your vision. That's what's called your peripheral vision. That's away from the fovea, so that's going to be out away from the sides of your head in the extremes. That's you can detect movement and you can detect a kind of shape in the periphery, but you cannot focus very well, and you cannot detect color with the rods. There are many less cones in the retinal wall than there are rods, but they help you see in color. They help you see more definition and detail of the objects that you see. It helps you to see in higher amounts of light, like daytime or bright light. They are clustered or centered around the focal point of the eye, or focal point of the retinal wall, which is the fovea. The explanation of why animals can see more of the visual field than we can, like predatory animals like cats, is they have a lot more rods and less cones, in some cases almost no cone activity at all, which allows them to see more movement and to see in much lower light, but they do not see very well in the daytime like we do. The spectrum of electromagnetic energy, which is basically just how we're measuring billionths of a meter, which is a nanometer. When you look at the detectable color or chromatic scale that the human eye can actually detect, the colors of the rainbow, if you will, that's from, let's say, about, I think it's 350 to 750, and that's in nanometers. That's 350 billionths of a meter, and that's the frequency of the wave itself. That's what can be detected by the human eye. So you can see gamma rays, X-rays, UV rays, infrared rays. So UV rays would be subliminal for us, below our visual cliff, or I'm sorry, our visual threshold, and infrared rays would be superliminal. They would be above. And then radar, broadcast bands, and alternating currents. When you look at the wavelengths, you have to look at amplitude and frequency. So you have things like short wavelength, high frequency, which are going to be more blue colors. You have earthier tones, which are going to be reds and browns, oranges. Those are going to be longer, slower wavelengths. You have greater amplitudes, which are brighter colors. So the higher the peak and lower the valley of the wave itself, it's going to be a brighter color. It's also going to be a faster frequency. Usually if it's a brighter color, so if you, let's say for instance really bright yellow, it's going to be a fast frequency, let's say a high frequency, great amplitude light wave. Small amplitude light waves are going to appear more dull. Theories for color vision, why we see color, or what the color spectrum is. There are several theories. There is the classic television theory of RGB, and that's not based on television, but that's every original television screen only had photocells for red, green, and blue, and they made every other color out of those three, and so that would be the trichromatic scale, or the 3 colors. That's the idea or the theory that the retina has 3 different color receptors, that you have cones that are sensitive to reds, cones that are sensitive to greens, and cones that are sensitive to blues, and when stimulated they can produce the perception of everything in between those 3. Does not explain or account for yellows. So it's a good explanation for color deficiency or color blindness. When you look at this, just basic colorblind test here on the right of your screen, if you can't see the green number 74 inside of that, it's because you are not very sensitive to the color green, which would support the Young-Helmholtz theory, or the trichromatic theory. Tri meaning 3, chroma meaning colors. Opponent process theory is not how many colors we can detect, but it's this theory or the idea that every color has an opposing or opposite color. So you understand red as an opposition to green. Your brain can perceive yellow as an opposite to blue. That's not a yellow word, I just realized there. And your brain can perceive white as an opposing color to black. So every color has an opposite or an opponent color. And again, this explains the after image effect, which we're looking at here with this flag, the British Union Jack there, that if you were to stare at that and then stare at a blank white screen, you would see the opposing or opponent color. That's the opponent process theory. Now we get into sound. This is the auditory cortex in the brain, which is in the temporal lobe. This is the surface of the brain that is around the temples, just above the ears. The auditory cortex is one part of the temporal lobe. You also have Wernicke's area in the temporal lobe, which allows you to understand and process speech that you're hearing or reading or seeing. And obviously, if you're seeing it or reading it, it's going to involve other parts of the brain, like the visual cortex. But audition is perceived in the auditory cortex, which is in the temporal lobe. When you look at the parts of the auditory process or audition, you have the outer ear, the middle ear, and the inner ear. Those are the 3 kind of regions of the audition process. So sound waves, they move the eardrum, which is the tympanic membrane, which stimulates the bones, the smallest bones in your body, the hammer and the anvil and the stirrup, and they vibrate the cochlea, which is a fluid-filled spiral sac on the inner ear where transduction of vibrations into sound waves actually happens. So getting into the sections, the outer ear, the middle ear, and the inner ear. The outer ear is the external ear itself, collects and concentrates the sound waves. That's what your ear tissue does, your actual ear, and the ear canal concentrates the sound down to the eardrum. And then you get into the middle ear. That's the bones, the tiny bones in the middle ear. They vibrate against the cochlea. And then you get the inner ear, and it's pretty easy to think of the sensory neural part of your hearing. That's where transduction occurs. So once you get vibration of the cochlea by the stirrup, then basically you get this, the nerve cells that start to convert these vibrations into pulses. And the auditory nerve, just like the optic nerve for your eye, the auditory nerve for your ear is going to bring sound vibrations as waves from the cochlea to the auditory cortex. And again, it's going to pass through the thalamus, as you can kind of follow visually in your mind the track of this sound wave as it goes through the ear process. It's going to go through the limbic system right about the thalamus and then travel to the opposite auditory cortex on the other side of the ear. So you can get a better idea of kind of how the thalamus is involved in all of this. It's kind of right in the middle of your head in your brain, so that it catches and routes all of this sensory data to its destination. So those are the 3 regions of the auditory process. How does it work? How does pitch work? Well, pitch is involved in the little tiny vestibules in your ear, the little tiny hairs that you have, and you have vestibular sacs and things like that in your ear. So we have theories like place theory. We hear different pitches because different sound waves trigger activity at different places along the cochlea, along the basilar membrane. And the basilar membrane was used in a FRQ fairly recently, and it was the most missed point on that FRQ. Most students who got points, or who lost points, lost that point because they did not know what the basilar membrane is. That is a part of the cochlea that has the little hairs in it that's involved in pitch, understanding pitch. So let's get into frequency also. Brain reads pitch by monitoring the frequency of neural impulses. So in other words, how often, not just where, not just the place of the sound wave, of where it stimulates in the cochlea, but how often does the sound wave travel across that membrane, those little hairs. Hearing loss, we have two different types of hearing loss. This is also common in FRQs. We have conduction hearing loss and sensorineural hearing loss. Conduction hearing loss is if the eardrum is punctured or the tiny bones of the middle ear lose their ability to vibrate. This is something that happens with damage. Sensorineural hearing loss is something that it's damage to the outer or middle ear. Sensorineural hearing loss is damage or malfunction of the neural processes in the inner ear. So that's the difference. Conduction affects the outer and the middle ear, and sensorineural hearing loss affects the inner ear, the cochlea itself. Transduction cannot happen, or the auditory nerve. So nerve deafness, it's caused by disease, heredity, aging, and prolonged exposure to ear-splitting noise or music. Again, you can kind of see how it works. You have the eustachian tube, you have ear tube drains excess fluid into the outer ear, and the middle ear bones can be seen once the fluid is cleared. Conduction hearing loss obviously is damage that happens to the outer or the middle ear. The sense of touch, again, there's only certain profiles of touch that we can detect. When something lands on the skin or applies pressure to the skin, you can only detect temperature differences, the amount of pressure that's being applied, or a pain signal. Those are the only things that you can detect: pressure, warmth, cold, and pain. So again, there is a cortex of your brain, just like there is for audition, the auditory cortex, which is in the temporal lobe. That's where you process sound. You have the visual cortex and the occipital lobe in the back of your head, where you process sight. And you have the sensory cortex at the top of your brain and the parietal lobe, and that's where you detect a sense of touch. Sends sensory information to the somatosensory cortex of the brain. The sense of taste, chemical sense that stimulates the taste buds, which send neural messages to the brain. And again, this is getting a little bit more chemical, much like the sense of smell. But unlike the sense of smell, it is still a thalamic process. It is still a process that goes through the thalamus because it is a process that transduction still has to occur. And the taste profiles are sweet, salty, sour, bitter, and this protein taste, umami. Olfaction, which is the term for smell. You have an olfactory bulb. You do not have an olfactory cortex. There's not a cortex of the brain that's involved in smell. Therefore, the thalamus does not get involved. It is an actual chemical reaction which happens when the mucous membranes and the olfaction in the olfactory bulb detect odorants in the air, or they do not. So whether or not something has a detectable odor depends on whether or not those scent molecules can activate the mucous membranes in your nose. So again, it goes through the olfactory nerve. It does not have a cortex in the brain like the other four senses. Kinesthesis. Kinesthesis, again, kina or kines means movement. So anytime you see kinesthetics, the kinesthetic sense, kinesthesis, kinesthesiology, that's body position and movement. Okay, typically it's going to be movement. So just like you have pain sensors in your nerve endings, you also have sensors in various parts of your body that send signals back and forth to the brain. So this is going to be sensory information that comes in. So these are going to be sensory inbound messages to the brain. They're going to have to go through the spine, and these are going to be joints, tendons, bones, ears, skin sensors, and obviously this is going to include nociceptors, which are pain receptors in the skin. And this is going to be the messages that come to your brain to tell you where your body parts are. So how your brain can control them is to know relatively where they are. How do we know where our feet are? We can feel the pressure of the messages going to the brain that your feet are below your knees. You're standing upright. The feet are below you, and your brain knows exactly where your body parts are. Your brain can detect your hand even when it's behind your back. I know a lot of you probably just put your hand behind your back so that you can kind of simulate what we're talking about here. But even if you can't see your body parts, your brain can anticipate where your body parts are. So that's what kinesthesis is. Sensors in your joints, tendons, and bones and ears, as well as your skin, send messages to the brain about where the body parts are located. The reason why you can walk without watching your feet or sit down in a chair without watching is that your brain perceives where those body parts are. Vestibular sense, again, you have vestibules or vestibular sacs in the inner ear. You look, they're kind of C-shaped. You have right and left vestibular sacs in the inner ear, and that's what tells your brain what position you're in. If you think about a liquid level, like a level that you can use to put on a surface to see if it is level or not, it has a bubble in it, and there's probably two lines that are scored on there, and what you're looking for is for that bubble to be even. It's not to one side or the other side. That's essentially, metaphorically, what happens in your brain for your body position. You have these inner canals in your ear, and what they do is the fluid, and where that fluid is located, gives your brain a sense of what position you're in. So when you're standing upright, you should get a level feeling in the head, or in the brain, excuse me. So your brain knows that your body position is upright. If you lay down on the side of your head, say on the left side of your head, well, now that fluid has shifted and moved, and because of that fluid movement, your brain can detect that you are now in fact laying down and you're not standing upright. If you were to lay flat, bend over flat, lay on your back, lay on your right, lay on your left, the fluid shifts in your brain much like a level, but that bubble can detect that your body position has changed. Think of it that way, and that's an easy way to remember it. Pain signals, again, pain signals are separate signals that come from body parts, skin cells, and other various cells in the body have pain receptors. Your pain receptors are known as nociceptors. They are important to the central nervous system because they can tell you when tissue damage has happened by sending alerts to the brain. Pain is a very, very effective way to get you to stop doing whatever it is that you're doing so that you don't cause further damage. But pain also is more than biological. As you look at this chart from the book of the biopsychosocial impact of pain, you also have the top-down processing of expectation or fear, like the child who's sitting in the dentist's office there, and the psychological influences in the top right. There's attention to pain, that signal detection that we talked about in that first couple slides. Remembering that things are painful since the last time that you had a procedure done, it causes that expectation, which can influence the level of pain that you experience. Your mental state can influence the amount of pain that you perceive. Your state of sympathetic nervous arousal can impact the level of pain that you experience. There's lots of things that you can, people being around you, if you get injured in practice and you don't want to look soft, so to speak, or if you don't want to look weak, you may kind of suppress those pain reactions until later. We also know that there is gate control theory in the spine, essentially what endorphins do, blocking the reception of pain messages from the damaged site to the brain so that the brain cannot perceive pain. And the theory behind that is that there are times when you need to be able to escape danger or to get away from a threat. And of course, if pain is effective at getting you to stop doing what you're doing, it's not very evolutionarily sound for you to stop walking or running away from danger, even if your leg is broken. So adrenaline, endorphins, and gate control theory can explain how sometimes we don't feel pain. Those messages are blocked. They don't make it to the brain until later when that parasympathetic nerves kick in and calm us down. Perception. Perception is definitely something that's fairly convoluted, unlike sensation, in terms of the idea that there are different ways that we interpret the data around us. So it's the understanding of what we have seen with our eyes or heard with our ears, what we've smelled, or what we've tasted. It's how your brain makes sense of it. So here's some perception terms for you, some vocab terms. Gestalt psychology is basically what your brain does with pieces. It's an organized whole. The tendency to make sense out of various continuous pieces and make them into a whole. There's figure ground and foreground. So we have figures in the background, figures in the foreground, and figures against the backdrop of the background. Depth perception, again, that's a binocular cue that you get from both eye signals. You can make a dimension and depth from the shape of items and how far away they are from you based on binocular cues from each eye. You have the visual cliff, and the visual cliff experiment was the clear glass top table that they put infants on, and they put the same color material underneath the glass as was on top of the glass, but infants could still detect when they reached the cliff or the edge. Even though it was a solid surface, they knew that it ended. So it proved the theory that even when we are young infants, we have the ability for depth perception, that it's built into our DNA at birth. Binocular cues obviously would be the opposite of monocular cues. Monocular cues would be a message from each eye. Binocular cues is what your brain does with the messages from both eyes at the same time. Retinal disparity is the explanation of how, as humans, our eyesight is offset a bit. There is a disparity between what our right visual field and our left visual field bring in. There's definitely a disparity from our right eye to our left eye. Our visual fields are offset slightly so that our fovea is several feet in front of our face and not right in front of our face, as if both of our eyeballs and visual fields looked straight ahead. Grouping principles. This is what your brain does as a tendency to find patterns in a world that doesn't typically make sense. Even nonsensical things, your brain will tend to group them together. For instance, proximity. If you look at the top left here, you have 6 line segments there, but your brain sees 3 columns, 3 separate divisions, or it sees 2 spaces. But your brain has a tendency to group things together by their proximity. It also has a tendency to group things together by their similarity. Right? We have patterns here. This is not random according to your brain. Whether you see it in rows or columns, your brain perceives it as a part of a pattern and assumes it is continuous until it learns otherwise. Which brings us to continuity, the fact that this looks like a continuous pattern that flows on for infinity in both directions. So that's something that your brain assumes. It doesn't assume that this is a stopping point in a line segment, like this is some wire. It assumes that this is a looping wave that just continues on in both directions, even though we can't see it. And then the closure principle, the fact that your brain would perceive this as a triangle, even though it is not connected, it's just a bunch of line segments together. It detects the acute angles and sees a triangle from past experience. So grouping principles are all types of top-down processing, your brain's expectation. If you look at connectedness, again, your brain's past experience tells it that this is connected, these 2 dots are connected, and these 2 dots are connected, and they are in fact separate. But really, there's no sense to that. It's nonsensical. They are a-patterned. Examples of monocular depth cues. So these are things that you get from just one eye. And then your brain would take messages from each eye and put them together to make a whole. So you have the horizontal or vertical illusion. That's a monocular cue. Light and shadow effect. That comes from each eye. Interposition. The fact that this king of spades is behind the king of clubs because your brain's top-down processing and past experience tells it that it is. You have linear perspective, and this is where we get some of these optical illusions. The fact that this monster in the tunnel should be much larger than this monster because he's much closer to us, or the fact that this one should be a shorter, smaller line segment or bar than this one because this one is much farther away and takes up more of the road. That's an example of a monocular depth cue. Relative motion, the fact that if your eyesight is fixated, it can determine the fact that things are going to continue to move past you at the same rate and at the same speed and the same size. Relative height, that this is taller compared to this block here, or however you want to call those right angles, because it's a comparative statistic of monocular cues. And relative size, and you get something that's known as the phi phenomena. Is the detection of movement. If you study something long enough and your brain starts to perceive that as having movement to it. So if you stare at this, your brain assumes that this is kind of the zooming in, we'll say, of something that's busier, and it tends to move in your mind's eye. Constancy, this is another example of top-down processing where your brain can be fooled from past experience and tricks of perception. Shape, size, lightedness, and color do not change, which is an example of constancy because of movement, distance, or lighting. So example, as we know that tile A is the exact same shade as tile B, and even knowing that, we still can't see that that's the case because your brain perceives constancy, that this is in shadow when in reality these are darker. These tiles here are darker. Your brain also perceives this as not being the same dimensions as this table when in fact they are exactly the same. And the same thing is true here with the doors and the door size. We perceive those as being open. This door is more narrow than this door. And finally, extrasensory perception. These would be things, again, this would be above our sensory interpretation of what we could do beyond the five senses. Things like telepathy, that would be communication without verbal or nonverbal cues. It would be an example of communicating with people using nothing other than your mind's perception. Precognition would be to predict things before they happen. Clairvoyance would be able to see or perceive something that's happening well outside of your visual field or your auditory field, so you can focus and put yourself in a time and a place that was way distant from this one, and see something that's going on in real time in a totally different part of the world that's well beyond your sight or your sound view. That's clairvoyance. And psychokinesis, of course, would be kines, again, as movement, the ability to, and psyche as mind. So the ability to move objects using your mind. So create opposing force in the physical realm using some kind of message from your mind. That would be psychokinesis. So that's Unit 4. That would be your sensation and perception and all of the vocab terms that go with it. The next one that we'll post that you can stay tuned for is States of Consciousness, which is Unit 5. It's a pretty interesting chapter. It's also what I commonly refer to as the Drugs and Dreams chapter.